next, for the sake of time, we're going to move on. We're moving to the early investigator section of our session. And first, I'm going to welcome Dr. Julia Plank. She will talk with us about brain findings.
Okay, wow, I'm really nervous to follow these incredible talks by researchers and by families, but nevertheless, I'll move on, partly because of nerves and more importantly, because I'm really hungry. Okay. So yeah, I'm going to present on the T1-T2 ratio as an indicator of cortical myelin content in youth with RASopathies. So first of all, what is myelin? So myelin is a fatty substance that essentially forms a sheath around the axons of brain cells or neurons. And so myelin essentially acts as insulation, allowing for efficient neural signaling, and it's really important for effective communication between different brain regions. So there were three main reasons why we wanted to investigate myelin in youth with RASopathies. So the first is because previous work has shown in animal models that of Noonan syndrome and neurofibromatosis type 1, that there are myelin alterations in these syndromes. The second reason is that myelin has been frequently linked to cognition, and we often observe some cognitive difficulties in youth with RASopathies. So we were wondering if we might observe myelin alterations in these groups that could then be linked to cognition. And then finally, myelin is increasingly recognized as a potential target for treatment. So if we could target myelin, so myelin can change in response to experience, so myelin could potentially be represent a target for interventions aiming to alleviate cognitive difficulties. So before we can jump to treatments, we first need a way of measuring myelin content in youth with RASopathies. So to do that, we use the T1-T2 ratio. So this approach has several benefits. First of all, it is a non-invasive MRI technique. Second of all, this T1-T2 ratio uses frequently collected standard clinical scans. There's minimal post-processing required, which I really like as a data analyst. And previous research also shows that the T1-T2 ratio captures established patterns of brain myelination in other populations, and so potentially could serve as a proxy for myelin content in RASopathies as well. So our main research question is then, can an MRI technique, the T1-T2 ratio, identify differences in myelin content in youth with RASopathies compared to typically developing peers? So for our methods, we had 112 children come into the lab. 86 of these kids had RASopathies, either Noonan syndrome or neurofibromatosis type 1. And then we also had 26 typically developing peers. The average age in both groups was around 10 and a half. And all of these kids kindly completed an MRI scan for us. So we acquired T1 weighted and T2 weighted images, which again are very standard structural images. And in total, acquiring both of these scans took around 10 minutes. So in terms of processing the images, there were 5 main steps. First of all, some corrections for motion and distortions. Second of all, dividing the T1 weighted image by the T2 to form the ratio. And that allowed us to get these really beautiful images. This is an example from one of our participants of their beautiful T1-T2 ratio maps. And then for our analysis, we selected 68 cortical regions across the brain. So this is the outer surface of the brain. And then within each of these 68 regions, we calculated the average ratio within each region. And finally, we wanted to compare the regional T1, T2 ratio between our two groups, so between RASopathies and typical developing kids. And for our statistical analysis, we did an analysis of covariance, including covariates of sex, age, curvature, cortical thickness, and also an index of motion. So before getting to the more interesting results, in my opinion, I wanted to look at the average signal distribution. So these are two representations of the average signal in the RASopathies group at the top and the average signal in the typical developing group at the bottom of the T1, T2 ratio across the brain. So we can see that there in the areas that are yellow or orange, there's more T1, T2 signal, which is indicative of more myelin content compared to other areas. And we can see that in areas like the motor strip, as well as the visual cortex and the occipital lobe, that there appears to be more myelin content, which is consistent with what we would expect from other research, that these areas are generally more myelinated than others. And the other thing that we can see here is that the typical developing brain appears to be slightly brighter or more yellow, indicating that there might be higher myelin content in the typical developing group compared to the RASopathies group. And indeed, that is what our statistical analysis showed as well. So when we compared each of the 68 regions between the typical developing group in blue and the RASopathies group in gold, we found this really clear pattern where the average T1, T2 ratio in the typical developing group was consistently higher than in the RASopathies group. And our statistical analysis showed that this was significant in 63 of the 68 regions following a false discovery rate correction. So overall, these results showed that there seemed to be less myelin in children with RASopathies compared to typically developing youth. So we also looked at subcortical myelin. So we looked at 8 subcortical regions as well. And we found a significant difference only in the accumbens, which is shown there in dark blue, where again, we found a slight reduction in myelin content in the RASopathies group compared to typically developing peers. Okay, so overall, the main takeaway from this is that cortical myelin appears to be reduced across the cortex in youth with RASopathies. We don't know exactly why this is, but it could be to do with disrupted cell migration of neurons or myelin-producing oligodendrocytes from the white matter to the cortex, leading to reduced myelin in the cortex in youth with RASopathies. And finally, I didn't present on relationships with cognition today, but I can tell you from our work and from previous studies as well, the relationship between the T1, T2 ratio and cognitive outcomes still remains a little bit unclear. So further work is needed to assess the clinical relevance of these findings and understand how the T1, T2 ratio relates to behavioral and cognitive outcomes in these populations. So yeah, thank you so much for listening, and a special thank you to all the participants in this work. And we are also currently recruiting adolescents with NF1. So yeah, thank you.
Thank you. Questions? Jayshree.
Beautiful talk. My question relates to longitudinal assessments and the relationship to age. And in your cohort, was there a specific age range for both the unaffected and children with a RASopathy diagnosis? And do you plan to do a follow-up at some point to see if there's quote unquote catch up? And then the last thing I want to say is just as a clinical manifestation, our whole blanket term of hypotonia, we suspect has something to do with the rate of myelination. We tend to think of that as peripheral, but I wonder if that could also be central.
So the first question regarding age, so the age range of our participants was the same in both groups, and it was a broad age range. So it was from age 6 up to 16. So we did also look at like different age bands to see if we still observed those differences, which we did. The second comment around a longitudinal approach I agree, that's much needed. And if someone wants to give us funding, that would be great. And then the third point, that sounds really interesting, and I don't know anything about that, so I'll have a look into that. Thank you.
Rini?
Fantastic talk. Thank you. Just to kind of piggyback on that last question about this question of, is this delayed myelination versus sort of kind of chronic undermyelination? Have you thought about looking at the different, it looks like you had a region of interest kind of approach, looking at earlier, like corpus callosum versus like later myelinated tracts, and sort of comparing across tracts to see, are there some tracts that are more matched than others?
Yeah, so this study was focused only on the cortex and the gray matter because the T1, T2 ratio seems to be more valid in the gray matter than for white matter. However, we did use a different approach and look at the white matter using another MRI technique, quantitative T1 mapping, which actually indicated greater myelination in kids with RASopathies in the white matter. So yeah, I think your question is a really interesting one, and definitely one that we are thinking about, and my next area of interest is trying to bring all these findings together and see if we can better understand if it's precocious myelination in one area or general delayed myelination, and trying to bring these different findings together, recognizing that each MRI technique itself is like a proxy for myelin content and not totally specific, but bringing them together, we might be able to reach some more specific conclusions potentially.
No more questions? No? Okay. Any more questions?
Very interesting talk. I just have a very quick question. Have you looked at actual cortical thickness and see if there are actual differences in the cortical thickness that, getting at your, the, neural migration or other related?
Yes, there are definitely differences in cortical thickness, which I think another person in our lab has published on. So I did look at the differences in cortical thickness also because that can also be considered a proxy for myelin content. But these differences go beyond the differences in cortical thickness, which is why I included it as a covariate in our analysis.
We also look on 3 different cortices.
That's true.
Maybe you can tell him.
Oh, okay. Yeah. We also looked at 3 different depths of the cortex as well, because we know that there are different different compositions at each level of the cortex. So we looked just at the gray-white matter boundary, as well as right at the surface and at the midline. These results are at the midline. And we found the significant differences across all different depths.
Thank you. Thank you very much.